DNA double helix structure by Watson and Crick. This concept is very much common among us. From the very beginning, when we have studied, we have known that name DNA, RNA. We also know this is a very common name, that DNA model was Watson and Crick model, DNA double helix model. But prior to discuss about the detailed structure of DNA, we should also have give a tribute to that scientist. Work of those scientists give us that the detailed information we know about Watson and Crick model, DNA double helix. This two name is very common, Watson and Crick model. But not only Watson and Crick, we should have mentioned the others name of these scientists also. First of all, Franklin and Wilkins. Scientist Franklin and Wilkins. They used X-ray diffraction technique. X-ray diffraction technique said that it is a DNA double helix based on the photograph from X-ray diffraction technique. Watson and Crick produced the famous double helical structure of DNA in 1953. In 1962, for this work, in 1962, Watson, Crick and Wilkins, the Nobel Prize was given to them. Watson, Crick and Wilkins. Before the Nobel Prize was awarded, Wilkins died because of cancer only in the age of 38 years. So prior to discussing the structure of DNA, it was our tribute to those scientists. So nucleic acids. Let's concentrate about nucleic acids. Nucleic acids, which are of two types, DNA and RNA. DNA is mainly found in nucleus and RNA is mainly found in cytoplasm, and RNA helps in protein synthesis. Main function of RNA is protein synthesis and it carry the genetic information. Carry genetic information which is transmitted from generation to generation. DNA, it is found in nucleus, but it is also found in mitochondria and this DNA is called mitochondrial DNA. DNA is also found in chloroplast. Okay, so this is three organelles: nucleus, mitochondria and chloroplast. And DNA in eukaryotes is long, unbranched and spirally coiled. DNA, when present in prokaryotes, it is circular. And DNA, when present in mitochondria and chloroplast, it is also circular. So, let's concentrate about structure of DNA. DNA, it is a polymer. Poly means many. When something is called that it is polymer, it is actually means that it is made up of many monomers consisting of long chain monomers. And these long chain monomers, these monomers are called nucleotides. DNA molecule, as DNA molecule consists of polymer of nucleotide monomers, so it is also called polynucleotide. Three components of nucleotides: sugar, pentose sugar, nitrogenous and phosphoric acid. Nucleoside, here we have another term, consists of only pentose sugar plus nitrogenous bases. Nucleoside, and nucleoside plus phosphoric acid it is known as nucleotides. Side S is being replaced by T. Nucleoside, when attached with phosphoric acid, it becomes nucleotides. Here we are going to study detail about structure of nucleotide. Pentose sugar, as the name suggests, pentose, penta means 5. Pentose sugar are of two types present in DNA and RNA. In DNA, in RNA, ribose sugar is present, and in DNA, deoxyribose sugar is present. Deoxy. So first of all, we are going to draw ribose sugar, and then I will show how from ribose to deoxyribose sugar is formed. Let's draw. Pentose sugar. It is a pentameric structure. Pentameric structure. This is a pentamer, and here is carbon number 1, carbon number 2, 3, 4 and the fifth number of carbon. Fifth number of carbon is present here. Now let's draw H, OH, here also H, OH, here also H, OH, here also H, CH2 and OH. Now let's draw here carbon. One, here will be carbon, 2, here carbon 3, here carbon 4 and this one is 5th carbon. So let's write the formula. C5 and how many H is present here? 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So C5H10 and how many O is present here? 1, 2, 3, 4, 5. C5H10O5, this is pentose sugar and this is ribose sugar which is present in RNA. Now deoxy, anything from D means minus. Okay. So deoxy means one oxygen will be eliminated. Now let's see which one oxygen will be eliminated here. This oxygen attached with carbon number 2, this will be eliminated here. This is ribose sugar and let's draw again deoxyribose sugar. This oxygen will be eliminated. This oxygen will be eliminated. So this one is deoxyribose sugar which will be present in DNA, and ribose sugar, this will be present in RNA. C5H10 and be careful, just remember about this 2 carbon. Here O is out, so a single O, O4, and this is deoxyribose sugar. Note that there is 2 functional group present in RNA, here ribose sugar, and here a single functional group is present and this functional group is not present here. And for this 2 functional group, RNA is very much active. For this 2 functional group, RNA is very much active and unstable. And for this single group, DNA is not so much active and stable. It is monosaccharide sugar. This is pentose sugar and this is monosaccharide, and mind it, 2 carbon position, from 2 carbon position 1 oxygen will be eliminated out to make deoxyribose sugar. It is from 2 carbon from which 1 oxygen is eliminated to make ribose sugar to deoxyribose. Now comes to the next constituent of nucleotide, and that one is phosphoric acid. Phosphoric acid. It is phosphate radical when present in aqueous condition, aqueous media. Phosphoric acid in aqueous media give rise to phosphate radical. Let's draw the structure. Phosphate have valency number 5. First two valency occupied by oxygen and another 3 occupied by OH radical. When in aqueous condition, this is H3PO4. This is structure of H3PO4. Yeah, phosphoric acid. Phosphoric acid when present in aqueous medium. This aqueous medium, phosphoric acid has tendency to lose this H. As it is acid and in aqueous condition, its tendency is to give out these 3H, and you can written is as O minus. This H O minus, you can written is as O minus. So this is structure of phosphoric acid. Now we will going to discuss about the third constituent nucleotide. That one is nitrogenous bases. Nitrogenous bases. There are two types of nitrogenous bases found here, purine and pyrimidine. Purine and pyrimidine. There are two types of purines are found here, and there are three types of pyrimidines are found in nucleic acid. Two types of purine is adenine and guanine, and cytosine, thymine, and in RNA uracil. And in DNA thymine. Okay, adenine, guanine, cytosine, thymine and uracil. Point number 2. Purine is dicyclic and pyrimidine is monocyclic. Here let's draw the structure. Pyrimidine is single ring compound and this is double ring compound. Let's draw pyrimidine first. Aromatic, heterocyclic, compound. Heterocyclic means as C is present along with N. That's why it's called heterocyclic compound. Now let's draw pyrimidine structure. As we know, the valency of carbon is 4. Now let's balance this. Mind that this one is number 1, N1. This is N2. And this is position number 2. This is first carbon and this is second carbon, and here carbon position 2, 3, 4, 5 and 6. This one is pyrimidine ring. Let's draw the purine ring. And here one, 2, 3, 4, 5, 6, 7, 8, 9. This is parent compound. This one is also parent compound, and this is imidazole ring. This is imidazole ring. 4 nitrogen atom is present. Point number 3. 4 nitrogen atoms is present here, and position 1, 3, 7 and 9 position, and here 2 nitrogen atoms are present, and the position 1, first and third position, and here ninth nitrogen bond will make bond with first carbon of pentose sugar, and here in case of pyrimidine this nitrogen, first nitrogen plus first carbon of pentose sugar. So this is all about nitrogenous bases, okay, and this will be attached by glycosidic bond, also by glycosidic bond. So this is all about nitrogenous bases. After making the three subcomponents of nucleotides, now it's time to finally make a complete nucleotide molecule. Now let's draw. Here, at the time of discussing the nitrogenous bases, I have already told you that first position of carbon, first carbon of pentose sugar, nitrogen bases will be attached with first carbon of pentose sugar. So what is first carbon of pentose sugar? So here N2 bases will be attached. Okay, here N2 bases means 9th nitrogen and first nitrogen of pyrimidine base. Here the bond is known as glycosidic bond, and phosphate group will be attached here 2 carbon, 3 carbon, 4th carbon and 5th carbon is here. Here 5th carbon. Here 5th carbon. Okay. And the phosphate group will be attached with 5th carbon position of pentose sugar. Here H2O will be released and phosphodiester bond will form here. Now let's complete this structure. Pentose sugar, and this one is nucleotide structure. This one is the nucleotide structure. Final, let's draw some nucleoside and nucleotide, and let's make a table and look how table name is formed here. DNA and RNA. Nucleoside, nucleoside, nucleotide. Nucleotide. What is the nucleoside meaning? Deoxyribose sugar plus base, deoxyribose sugar plus base and here ribose sugar plus base. Add nucleotide, deoxyribose sugar plus base plus phosphate group. Here also ribose sugar plus ribose sugar plus base plus phosphate group. Okay. Purine base. Purine base. And here pyrimidine base. Okay. Purine means adenine and guanine. And what will be the name? What will be the name? Deoxy adenosine in case of nucleoside and deoxy adenylic acid or deoxy adenosine monophosphate. Here in case of RNA, when purine base is here, adenine, and here in case of adenylic acid, or adenosine monophosphate. Is it clear? And now let's see what's about guanine. Deoxyadenosine, DAMP, here adenine, capital adenosine monophosphate. Now deoxyguanosine, DG, deoxyguanilic acid or deoxyguanosine monophosphate, DGMP, here guanosine, G, and guanylic acid or guanosine monophosphate or GMP. Is it clear? The naming pattern. Now pyrimidine base. Here pyrimidine base. Cytosine, thymine and uracil. What will be the name here? Deoxycytidine or DC here, and deoxycytidinic acid or deoxycytidine monophosphate, and here cytosine and cytidinic acid or cytosine monophosphate or CMP. It's about cytosine. And what's about thymine? It will be present as deoxyribose only. So deoxythymidine or DT, and here deoxythymidinic acid or deoxythymidine monophosphate, DTMP here, and it will be present in ribose only, present in ribose only. So what will these both will be blank, and uridine or uridylic acid or uridine monophosphate or UMP. So it's all about name of the bases, nucleotide components found in DNA and RNA. So it's all about structure of a single nucleotide. In my next class, I discuss about the Watson and Crick model of DNA and how the DNA polynucleotide chain will be formed. Will be discussed their detail, and structure will be discussed their detail. So keep in touch.